clearly not going to win a gold medal in Olympic gymnastics. That's okay, because I'm not training for a gold medal in gymnastics, I'm training for a PhD in biochemistry. So today I want to tell you more about what my training does involve, which is studying this protein called Argonaute, or AGO, and how it's involved in this process called RNAi, or RNA interference, that helps regulate the levels of various proteins in your cells.
Kind of like how you have a thermostat in your house to control the temperature, your cells have all these mechanisms to make sure that the levels of proteins are optimal for that cell.
And this might change over time, so your cell has to be able to respond. And one way it responds is through regulation with RNA interference, or RNAi, especially using microRNAs. And at the heart of it is this protein that I study called Argonaute. And so Argonaute is this little thing right here. So it's a protein. What he does is he uses these short strands of RNA to target recipe copies, so protein recipe copies, mRNAs, that have a corresponding sequence. And then it shuts down production of that protein. So basically, you might have heard a lot about mRNA lately because of the coronavirus vaccines. But basically, an mRNA is just a copy of a protein recipe. And so our cells actually make RNA all the time. It's like this intermediary between the genes, or the DNA version of the protein making instructions, and the actual protein that's made. So basically the gene is like held in this membrane bound compartment of the cell called a nucleus. And then it's kind of like the reference section of a library. You can't take things out, but you can make copies of them. And so the cell makes these mRNA copies of those, and it ships those out of that nucleus into the general part of the cell, the cytoplasm. RNA copying is called transcription, and then this mRNA goes out into the cytoplasm, and these complexes called ribosomes then use those instructions to make proteins from them. What RNAi does is it targets this mRNA and it degrades it. And so it does this in a sequence-specific way using these small RNAs, so like microRNAs and siRNAs. And these RNAs have like a sequence that corresponds to a sequence in the in part of those mRNAs. And so then it can target those mRNAs and cause their degradation. But it has to find and bind them and recruit cofactors to help out. And so the protein that helps them do this is this Argonaute protein. So it's actually going to hold that small RNA and use it as a guide to go find those corresponding targets. So sometimes I like to think of it as kind of like a self-driving car, where you plug in the address using the small RNA and then go find that mRNA and shut it down. So our cells use this microRNA mediated regulation all the time to control levels of various proteins in our cells. And we also can use it therapeutically to target specific mRNAs for proteins that might be being mismade or like made too much of, or that sort of thing. And I'll show you an example of that later. We can also use like it in the lab to like quote unquote knock down genes, so to reduce the expression of certain proteins, so reduce how much of those proteins is made. And because you're doing it in the sequence specific manner, all you have to do is like introduce that a small RNA, so like an siRNA with that sequence, and Argonaute can help shut down production of that protein. And it's not messing with the original gene, so it's not permanent. And so RNAi is really awesome, and Argonaute's really, really awesome because it's the heart of all of this. And so I study how Argonaute is regulated and how it regulates protein production. So now let's go into a little more detail and some graphics and stuff. Proteins are these little like molecular workers in all of your cells, and there's different types of proteins that can do different things. And the instructions for making those proteins are written in the form of DNA as genes. So genes are basically just stretches of a longer piece of DNA called chromosomes. So these recipes, in order to be made into protein, they're first transcribed into mRNA. So our messenger RNA copy of this DNA is made, and then that gets used by protein making complexes called ribosomes in a process called translation to make a protein. And I study a process that kind of puts a damper on that whole thing. See, the amount of protein you get is going to correspond in large part to the amount of mRNA you have, because the more copies of this recipe you have in circulation for the ribosomes to use, the more protein you can make. So if you want to decrease the amount of protein you're making, one way to do that is to decrease the amount of mRNA. And RNA interference is a way that can specifically downregulate the protein production of specific proteins by reducing the amount of mRNA for that protein. So, because it's doing the sequence specifically, it can reduce individual protein levels without affecting other protein levels. And unlike transcriptional regulation, where you're actually making fewer mRNA copies, well, the problem with that is that it takes time, and you already have all those mRNA copies that are already in circulation. So RNAi allows you to get rid of those circulating mRNA, so you don't make that protein anymore. And so how does it do this? It uses these small RNAs. So by small, I'm talking these are about 22 nucleotides long. So nucleotides are RNA letters, so it's about 22 letters long. And these are going to act as guides to as guides, and so they contain these sequences that are complementary to sequences in messenger RNAs. And so typically in the 3' UTR of messenger RNAs, which is the tail of the mRNA. So after the actual part that codes for the gene, so the part that has the gene, that codes for the protein, so the part that has the protein instructions. After that, there's more. And we call it the 3' UTR, the untranslated region, because it's not translated into protein. But that doesn't mean it's not important. And one of the reasons why it's important is because it has these binding sites for these small RNAs. And so, by binding sites, I just mean that it has a region of complementarity. So there's this region called this at least 6 to 8 nucleotides long in what's called the C region I'll talk about. And so, but there are actually there are a few different types of small RNAs. So the type that our cells use the most is called microRNA or miRNA. And so this is made from, we have genes that make this, but instead different microRNAs have different genes, but they don't get translated into protein. They get used as the RNA form. So they're a form of functional RNA. So there are different types of functional RNA, and this is one of them that functions as the RNA itself and doesn't just serve as an intermediary between gene and protein. Another type of small RNA is siRNA, or small interfering RNA. And this comes from exogenous sources, so outside sources of double-stranded RNA. And so insects, some other invertebrates, and plants, they use this a lot as a antiviral defense mechanism. So they take RNA from viruses and use it to target the specific virus, because the viral sequence will then match to the viral sequence. If they one strand of the viral sequence will match to the viral sequence, and then that can be used to target it. But we have evolved to have more complex immune systems with multicellular immune systems with all these signaling cascades and all this sort of stuff. And so we don't use our RNAi machinery for that, but we can it uses the same machinery I'll tell you about. So our cells are capable of using small siRNAs if you introduce them, such as you can do therapeutically, or we do it a lot in the lab to knock down specific genes, so to reduce the amounts of certain proteins that are being made temporarily. So this isn't like CRISPR. We're not removing the gene permanently. We're just reducing the amount of the protein that's being made from it, which is really great because it's not permanent, and you can do this to specific genes using the specific sequences. And so how does the specificity comes about is, as I hinted at, there's a seed sequence. So the 6 to 8 nucleotide long region at the 5' end of the small RNA. And so RNA, we talk about it having two ends, the 5' end, which is kind of like the starting end, and the 3' end, which is the ending end. So the 5' end has a free phosphate group, and the 3' end is a free OH. Just so you don't get confused if that you were wondering what that meant, but you need to worry about that now, except know that there's those two ends. There's the 5' end and the 3' end, and then you have the seed sequence, which is this specific region of that small RNA. And this region is crucial for determining which messenger RNAs get targeted. So typically, although there are some weird exceptions where there's pairing in different places, this seed sequence, as we'll see, is going to be really important. This is like crucial for getting repression, so getting the mRNA degraded. And so how does you have all these small RNAs floating around in these cells, you have all this mRNAs floating around in the cells, how the heck are they supposed to find each other? And so you can also have further pairing. So that seed region is like the main determinant, but you can also have pairing, so like especially in this 3' supplemental region. So remember, the 3' end is like the other end, so this is like towards the 3' end. And this 3' supplemental region is like 13 to 16, and as we'll see in the structures they'll show you, there's kind of like a supplementary chamber which allows for this sort of pairing, which can enhance the binding affinity, so how strongly it'll bind, with the help of AGO, which is this protein I want to tell you about. So siRNA. So microRNA typically has this like partial complementarity, where you have that seed region complementarity, full complementarity, and then you might have additional complementarity in other regions of the microRNA. With siRNA, you have this complete complementarity. And so this is going to be important because it's actually going to allow for a different form of degradation. It's going to allow for that mRNA to actually get cleaved as opposed to just like degraded. And so, like I was saying, you have these you have these small RNAs floating around in the cell. You have these mRNAs floating around in the cell. And how are they supposed to find each other? Enter the protein I study, Argonaute, or AGO. This protein is awesome. It binds to that small RNA, and it uses it as a guide, which is why we call that a guide. It uses it as a guide to find those 3' UTRs, so find those messenger RNAs with the corresponding binding sites. And so it can kind of like hop along until it finds a sequence with their seed sequence matches, and then it latches on, and then if it can bind in the supplemental region and stuff. If there's further pairing, it'll make more sense when I show you the structures later. But the basic idea is that once you bind, then you can get translational inhibition and mRNA degradation. So you keep that protein from being made and decrease the amounts of the protein that are present. So where does this small RNA come from? So the microRNA, it's transcribed from genes, just like a normal, well, quote-unquote normal gene would be, just like the genes for the proteins, except that it doesn't go to the protein phase, and it doesn't form a messenger RNA. Instead, you have this long precursor transcript that folds up into these long hairpin thing. And this is called the primary microRNA, or the pri-mRNA. And this is happening in the nucleus, so that's the membrane-bound compartment in your cell where all of the DNA is housed. And so what happens is it gets transcribed as this long hairpin, and then it gets bound by this microprocessor complex. And it's going to cut it into a shorter hairpin. And it's going to go into the cytoplasm. So the cytoplasm is like the general interior in your cell, where the ribosomes, so the protein-making machinery and all of that stuff is in there, and all the proteins and all of that stuff. And so this is where it's going to function usually. But it's not ready to be functional yet. So right now you have this hairpin thing, but we need to get that single short strand of RNA that I've highlighted in orange here. And so what's going to happen is it needs to get cut again. And so it's going to get cut by this other nuclease, so RNA cutter called dicer. And so dicer is like a molecular ruler and scissors in one. So it's going to measure and cut to form an RNA duplex.
with these overhangs. And so that's also where the siRNA precursors come in. So like double-stranded RNA can come in and then also get bound by dicer and get cut. And so either way, you end up with this RNA duplex that gets loaded into this protein Argonaute. And this forms the pre-RISC, where RISC stands for RNA-Induced Silencing Complex. So it's not ready yet though, so we still have that orange part hidden. So we have the sequence that needs to find the mRNA, and it's hidden by the passenger strand, so the other side of the duplex. So the RNA strands are now separate, but they're still, they're not, you have two strands, but they're like glued together still. And so Argonaute needs to kick off that passenger strand. And now you have the mature core RISC, where you have that sequence exposed to go find the matching sites. And so what happens once it finds a matching site? Well, that depends in part on the Argonaute protein and in part on the amount of complementarity you have between the guide and the target. So humans have 4 Argonaute proteins, AGO1 through 4. AGO2 is like our main Argonaute, and it has this capability of slicing fully complementary targets. So by slicing, I mean it has its own pair of scissors, and it can cut that target in the specific place in between nucleotides 10 and 11 across from in like the guide numbering. But this is only going to happen if you have perfect complementarity, like you would see with siRNA. This isn't going to happen with like our microRNAs, which is what our cells use to regulate genes. But if that happens, you have exonuclease chewing, it's leading to mRNA degradation and target repression, so less of the protein present. If you have partial complementarity, like we have with at least for human microRNAs, what happens now is you can't slice it. So what happens is Argonaute has to recruit helpers, and so it binds to this long scaffolding protein called GW182 that's kind of like this long spaghetti-y thing with these different binding sites for different things. So it binds to Argonaute, and it also pulls in this like decapping complex to remove the 5' cap, and then a deadenylation complex to remove the poly A tail. And so those are like the protective things that messenger RNAs have on their ends. And so basically mRNAs, our cells have lots of exonucleases that will chew up RNA that has free ends that's present in the cell. And this is important because it like protects us from viral RNAs and that sort of thing. But we don't want it to chew up our own mRNAs, so our mRNAs have like these cap and tail added, and there's also other reasons for the cap and the tail. But in order to, if AGO can like get these proteins to expose those free ends, then they'll get chewed up. And so all AGO has to do is, so it binds, and then it binds to GW182 and like pulls in these other helpers. And once you have those ends exposed, you can get mRNA degradation and translational inhibition and sequestration. And so and then AGO can release that target and then like do it again and again and again. So it's super awesome. And that's why I study it. And so just another analogy that I sometimes use to help understand how cool AGO is it's kind of like a self-driving car, where the small RNAs, so like the microRNAs, are kind of like addresses that you plug into the car. And then AGO will go to those locations and shut down the protein production. And the mRNA gets degraded, but AGO is unharmed and can do it again and again because it's really, really awesome. And what's cool about microRNA is, so we have lots of different microRNAs. So we have lots of genes for different microRNAs, and then different genes have different microRNA binding sites in their 3' UTRs. And often they have combinations with like multiple binding sites for different microRNAs. And then the same microRNA might have binding sites in multiple different genes. And so this way you can have this like combinatorial regulation where one microRNA is helping regulate multiple related genes, and then you can also have additional regulation by like having multiple different microRNAs target the same thing, that allows you to kind of like increase your specificity and that sort of thing, and kind of titrate the amount that you want so that you can have like more or less repression based on how much of the protein you want destroyed or whatever. And so our cells use microRNA mediated regulation to regulate like most of our genes, so it's pretty cool. But we can also use artificial RNAi as a therapeutic, as well as in the lab. And so an example of a therapeutic is this drug Givlaari. And so basically there's this disorder called acute hepatic porphyria, and it has this problem with an enzyme, so a protein helper molecule that's involved in heme synthesis. So heme is like that protein, or heme is that molecule that carries oxygen in your blood. And so they have this problem in making heme, so they get this buildup of these byproducts because heme is normally regulating this pathway. And so instead, so that pathway gets dysregulated and you get an increase in these like buildup of these toxic products. And so what Givlaari is it's this RNAi drug that actually, so it's like an siRNA, but it has this GalNAc conjugate. So basically it's this like sugary thing that makes it get taken up by the liver cells. And so this RNAi is going to take over the role of heme, and it's actually going to decrease the production of this protein. And so that's one example of an RNAi used therapeutically, and we use it all the time in the lab. Well, I don't personally, but a lot of people do, for the reasons we talked about before. But how does Argonaute choose what to regulate and when? This is like the key research. This is one of the key components of what I like, why I'm studying what I do. So basically, if you can think about, you have this Argonaute, and it's in this like sea of messenger RNAs. And so which does it target? Well, we talked about how part of it comes from the amount of complementarity you have between the microRNA and the mRNA. And so the key determinant, remember, is that 6 to 8 nucleotide long seed sequence. And then you can also have additional complementarity. And so that can increase the specificity and the strength and that sort of thing. Regulatory information can also be coming from Argonaute, and this is what I study. So what does Argonaute actually look like? Well, you might think, so looking at Argonaute, you might think it kind of looks like a duck. So my boss, my PI of my lab, Lemore Joshua Torres, she came up with this analogy where Argonaute looks like this duck. And so where you have those 4 different colors I have shown there. So those correspond to the 4 main domains of Argonaute. And so a domain is just like a part of a protein that we talk about. Like it's just kind of like you might have different rooms in your house. We can talk about different domains of a protein. So sometimes they're like structural domains, so they'll fold independently of the rest of the protein. So if I were to like remove the rest of the protein, it would still fold and act the same. Sometimes there's functional domains, which is like it has a specific function. And often it's a combination of the two. And like domains can happen, like shared domains between different proteins. Like really similar domains that have like the same thing for different proteins, and so you might see the same domain in different proteins, like this PAS domain I'll tell you about. And so basically you have those 4 main domains. The end domain, called that because it's at the end terminal, so like that end terminal end of the protein has the amino acid free or amino group free. Then you have this PAS domain, which is the head of our duck. And then the mid domain and the PW domain, and then you have these 2 linker regions, so L1 and L2, which are kind of like flexible regions that allow the other domains to move more to accommodate RNA when it binds. Speaking of that RNA, this is the structure of Argonaute bound to a microRNA called miR20. This structure was solved by Elado Kayam in our lab, who was a postdoc in our lab, who's a really great mentor to me, and so I'm really thankful to him. or I guess he was a postdoc and then he was like a senior scientist in the lab. So I didn't want to like down credit you a lot. Thank you. And so basically here you can see that the 5' end of the microRNA is held here, and a pocket created by the mid and pWe domains. And then the 3' end is held in the Pas domain. That pocket that's holding the 5' end is like super strong. Like it binds really tightly. Like it'll stay stuck for weeks. It's pretty crazy. Which makes it really hard to purify Argonaute in its RNA free form, because it binds to like RNAs in the cells when you're making it. So you have to use tricks like using like high salt and like cation exchange and that sort of thing to separate the RNA bound and the RNA free. So all the purifications aren't like super simple, and you don't end up with nearly as much Argonaute as you want. But anyway, back to our story. So you can see that the microRNA looks kind of weird here because it's like missing that region. That's because this is an X-ray crystal structure. So we talked more about X-ray crystallography before, but basically it's this technique where you get a protein to form these orderly crystals, and then you shoot X-rays at them, and then those X-rays like get scattered by the protein, and then you work backwards from the scattered rays to figure out where the different atoms were located. And so sometimes the strength of the signals for that, well, it's actually looking at like the electron density. So like the electrons are part of the atoms that are like this cloud around the central nucleus. But basically, so you go from this like pattern of spots called a diffraction pattern back to the electron density map, which is this like meshy blobby thing, and then you try to fit the atomic positions into that to get this like atomic model, which is like this sticky, curly, sheety thing. And that was sheet-y, as in like those beta sheets, the beta strands that form these like pleated sheets. But anyway, so when you do that, sometimes the electron density for a region isn't strong enough for you to actually put that atomic model in. So this was a full microRNA, but it looks like it's broken because that region, it didn't have strong enough electron density to model, which likely means that that region of the RNA is really flexible. But you can see that seed region, so at the 5' ends, that's held really tightly, that's like the seed region, and you can see it's kind of like pre-organized. So those bases, so those RNA letters are like sticking out and ready to go search for matching messenger RNA sites. And so Argonaute kind of like samples different sites so it can kind of like travel over mRNA, and it kind of has the first part of the seed exposed, and it kind of like hops along, and if it sticks, so if it's like a matching sequence, then what happens is it kind of latches on and it starts moving. So it starts exposing more of that RNA. So it exposes more. So this is the guide only and just like a different view. And it starts this is bound to a short seed only target. And so what happens is you get this movement of the Pas domain, so like of the head of the duck away from the other region, and this is opening up the RNA binding channel, and it's allowing more of the microRNA or siRNA, the small RNAs. It's allowing more of the sequence to be exposed so that's how you can get like further binding in the supplemental region, which is like that region that we were talking about before, how there was like that supplementary chamber. So you can see it in this structure here. You kind of have these two regions where you have this double pairing, and then in between them is a more flexible region like this bridge region. And so Argonaute's really dynamic, and it can undergo these movements, and it's pretty cool, and that's just one of the reasons why Argonaute's really cool. And there's even more about why Argonaute's really cool that I'm not allowed to tell you yet, but I promise that once I finally publish it, I'll probably do like a whole video and tweet thread and stuff, because I'm really excited because I've never had a published paper, and I really am excited to tell people more about my work, and yeah, so till then, here's intro to Argonaute and microRNA and RNAi and some really cool stuff. Hopefully you think it's cool, because I think it's cool, which is good because I study it all the time. Okay, gotta go. Bye.